All right, so these are the checkpoints we sort of skipped right over. And I'm gonna kind of just go through these one by one. So the first one, name 3 structural differences between the epithelial cells of the proximal and distal tubule. And then the structure of which tubule is more favorable for exchange. So what were some of the differences here that we mentioned? Does anyone remember? Differences of the proximal tubule epithelial cells versus the distal tubule epithelial cells. Any thoughts here? Okay, so I'll just go over these. I'll go over this one. So let's jump back here actually. We said that the proximal tubules had a couple different things. They had those those leaky tight junctions. Here we go. So those leaky tight junctions, they also had microvilli, which gave them this brush border, okay? They also were taller cells, right, which is more adapted for that absorption. And then they also had a lot more mitochondria. Okay, so the microvilli forming the brush border, the leaky tight junctions, which facilitated paracellular transport, and then the mitochondria, which provides the energy, but also the space. So taller cells, which allow for more space, more machinery for absorption. Okay. All right, and then the cuboidal, the distal tubule cells, have less mitochondria. They are square-shaped cuboidal cells. They have tight junctions. They don't have microvilli. Therefore, there's no brush border. Okay. The second part of the question here, the structure of which tubule is more favorable for exchange, that would be the proximal tubule, where we see unregulated exchange, right? So things can go freely through the cell itself. There's more space for that. There's more energy for that. Things can also go freely through the sides of the cell because there's more, there's less of that secure tight junction. All right, number 2, the clearance of which 2 substances can be used to estimate GFR. So which 2 substances did we mention that their clearance rate was a good indicator or estimator of GFR? Oops. Does anyone remember these 2 substances? Okay, inulin, excellent. Yes, inulin was one. Inulin was actually the perfect indicator of GFR because its clearance rate was 125 milliliters per minute, and that is the exact value of our GFR, or exact rate of our GFR. Someone is saying glucose, so not glucose. And if you could say why, if someone else could share why not glucose, that would be excellent, but not glucose. Glucose is not a good predictor of GFR because of a really important fact about glucose. But there was another substance, apart from inulin, that was a good predictor. Not as good as inulin, but it was a decent predictor of GFR. It was slightly more secreted, and so it had a higher clearance rate, but it was in the range of that GFR rate. Creatinine, yes, that's correct. So creatinine is another substance that is one of the breakdown products from proteins, and it is an excellent, not as good as inulin, but it's a good predictor of GFR as well. And it's excellent in that it's less invasive. So I want to make that clear. It's not excellent in that it is it's correct or adequate or accurate. Let me say it's not accurate in terms of that prediction, but it is good because it's less invasive. Our body actually makes this, and so we don't have to exogenously introduce anything into our vein. So it's a good predictor of GFR in that way. Okay. And then just to answer that side question here, glucose is not a good predictor of GFR because it is fully reabsorbed. And so there's no way to tell. It should have a clearance rate of 0, right? And so it's kind of the worst predictor of GFR because it doesn't get cleared. It doesn't come out in the urine. And so there's no way to see what the GFR, the filtration rate looks like because it doesn't get cleared, okay? So glucose is not a predictor of GFR. All right. Let's move on to the second part here. The clearance of which substance can be used to estimate renal plasma flow. So this is blood flow through the kidneys. So which of the substances did we say would be a good predictor of renal plasma flow? And if we're talking about flow through the vasculature, something that is a good predictor of flow in the vasculature would have to be coming from the vasculature. So kind of a small hint here, but this is something that has to be aggressively secreted. So coming from the vasculature, being moved into the tubules at a really aggressive rate, and not being reabsorbed at all, because that's not going to tell us about flow. If it's going back into the vasculature, it's going to do the opposite. We want something that's coming from the vasculature, into the tubule. And so that would be the best predictor of blood flow, of renal plasma flow rate through the vasculature. Okay, and so that was PAH, para-aminohippuric acid, or PAH. We said it was heavily secreted, not at all reabsorbed, and so it had a really high clearance rate, but that was good because that's telling us about the flow rate of things coming through the vasculature because it is so heavily secreted. All right, excellent. Let's move on to the third part here. So if the clearance of molecule X is greater than the GFR, was it reabsorbed or secreted in the renal tubule? So if I'm seeing a clearance rate of a molecule that is at a higher rate than my GFR, so greater than 125, is that being reabsorbed or is it being secreted? Secreted, excellent, thank you. Oh, excuse me. Okay, yeah, so that is absolutely correct, secreted. If it's being cleared faster than it's being filtered, it must be coming from somewhere else. So it must be secreted. If it is being cleared at a rate that's slower than it's being filtered, right? It's coming out less in the urine, then it must be reabsorbed, okay? And then the last question here having to do with micturition. So describe the innervation of the bladder, internal urethral sphincter, and external urethral sphincter. And so was the innervation of the bladder. So just to go over this in the interest of time. So the innervation to the bladder is the parasympathetic division of the autonomic nervous system. Innervation to the internal urethral sphincter was the sympathetic division of the autonomic nervous system. Innervation to the external urethral sphincter was the somatic nervous system, okay? And then the second part of the question, voluntary inhibition of micturition, in other words, guarding. So that guarding reflex through innervation to which of these structures? So voluntary inhibition of micturition happens through innervation to which of these structures? Okay, so let's just go over the answer here again. So voluntary inhibition happens to the external urethral sphincter, okay? Again, voluntary being the key word there. If I'm deciding to not go to the restroom, if I'm deciding to guard my bladder and not void, that decision, that voluntary component is the the innervation to the external urethral sphincter, which will contract that sphincter and really keep that tight seal, keep that muscle closed.